Hi, I'm Mai Pomato, and today I will be reviewing the topic of viruses, Chapter 19. I will be talking about what a virus is, their structure, different types of viruses, how they replicate, their replicative cycles, and more. So, a virus is defined as an infectious particle consisting of little more than genes packaged in a protein coat. While viruses are unable to carry out metabolic activities outside of a host cell or reproduce, researchers in the late 1800s proposed that viruses were the simplest of living forms. What is the structure of a virus? The smallest viruses are 20 nanometers in diameter, while the largest have a diameter of several hundred nanometers. They consist of a nucleic acid, a protein coat, called a capsid, and a viral envelope. Capsids are made up of many protein subunits called capsomeres. The viral envelopes surround the capsids of the viruses and help viruses infect their hosts. Viruses can contain either DNA or RNA as their nucleic acid. Viruses containing RNA are known as retroviruses. These RNA animal viruses contain a special enzyme called reverse transcriptase, which transcribes an RNA template into DNA. These viruses are called retroviruses because they cause an RNA to DNA information flow, which is the opposite of the usual direction. The word retro means backward, hence the name retrovirus. Another type of virus is a bacteriophage. These viruses are helpful because they only infect bacteria. The viruses contain capsids with heads that enclose their DNA. A protein tail piece with fibers is attached to the head, and these fibers allow the phages to attach to the bacterium. How do viruses replicate? Because they lack metabolic enzymes and proteins, viruses can only replicate in the host cells. With that said, each particular virus can infect cells of only a limited number of host species. This is called a virus's host range. Viruses can replicate by two alternative mechanisms, which are the lytic cycle and the lysogenic cycle. What is the lytic cycle? The lytic cycle is a phage replicative cycle that culminates in the death of the host cell. The lytic cycle begins with a phage attaching to a host cell and injecting it with its DNA. Then, the phage DNA circularizes. New phage DNA and proteins are synthesized, and these components created phages. Due to the vast replication of the phages, the cell lyses, and the phages inside are released. On the other hand, the lysogenic cycle allows replication of the phage genome, but without destroying the host cell. The phages that can use both modes of replicating are called temperate phages. Unlike the lytic cycle, the lysogenic cycle begins with a daughter cell containing a prophage. Then, the phage DNA integrates into the bacterial chromosome. This results in the creation of a prophage. So, the bacterium containing the prophage reproduces normally, causing the prophage to replicate and be transmitted into the daughter cells. Many of these cell divisions will result in a large population of bacteria that have been infected with a particular prophage. A prophage is the viral DNA of a bacteriophage, and it is able to produce phages. You may be wondering, how did viruses originate? Viruses have been found to infect every form of life. Most molecular biologists believe that viruses originated from naked bits of cellular nucleic acids that moved from one cell to another. Many viruses are very dangerous and can lead to the death of organisms. To prevent this, scientists came up with a medical tool that serves the purpose of preventing viral infections, called a vaccine. A vaccine is defined as a harmless variant or derivative of a pathogen that stimulates the immune system to mount defenses against the harmful pathogen. Although these vaccines can prevent certain viral illnesses, medical technology can do little to cure most viral infections once they occur. Viruses that suddenly become apparent are referred to as emerging viruses. An example of this is the AIDS virus, which appeared in the 1980s in San Francisco, out of nowhere. Another example is Ebola, which is a deadly virus initially found in Central Africa. Viruses can also appear in plants. Plant viruses have the same basic structure and mode of replication as animal viruses. A major difference is how these viral diseases are spread. They are spread in plants by two major routes, which are horizontal and vertical transmission. In horizontal transmission, the plant is infected from an external source of the virus, and the invading virus works to get past the plant's epidermis, which is its outer protective layer of cells. Herbivores, such as insects, can act as carriers for viruses and spread it from plant to plant. Lastly, vertical transmission is where a plant inherits a viral infection from a parent. This route of viral infection can occur in both asexual and sexual reproduction. For my final topic, I'm going to discuss the two simplest infectious agents. These are viroids and prions. Viroids are very small circular RNA molecules that infect plants. They do not encode proteins, but can replicate in the host plant cells. Viroids can cause abnormal growth development and stunted growth. Prions are infectious proteins. and they have been found to cause a number of degenerative brain diseases in many different animal species. One well-known example of this is mad cow disease. I hope this helped to further your knowledge of viruses. Thanks for watching.